Welcome to Trial Site News. We are excited to introduce our guest, Rich Horgan, whose story inspired us to write more about his accomplishments. In a story of brotherly love, a familial connection, and the power of drive, desire, and entrepreneurial energies, the Boston-based entrepreneur whose brother has Duchenne muscular dystrophy launched a biotech nonprofit in a quest to find a cure for his brother and others with this condition. So, Rich, first and foremost, thank you for joining us today on the Trial Site News podcast show.
Absolutely. Thank you very much for having me.
So, Rich, we know the story of the inspiration to help develop a cure to your brother. It was an inspirational story for us, and hence why we published an article on your story. Now, having a family member that you love very much that has a major health challenge, it's tough and it's a challenging thing, but this can also bring a family closer together. Could you share with us the events in life that led up to you thinking about launching a biotech organization? When did you finally internalize the need to actually start this particular biotech venture?
So, growing up, my brother was 4 years younger than I, and so unlike most kids who go outside and play baseball and basketball in the summertime or springtime or fall, my brother and I oftentimes resorted to playing video games inside or doing more activities within the house. And as a kid, you don't really realize the why behind that. But as I grew up and became a teenager, the why started to become much more obvious. My brother falling behind in growth curves, Terry not really just doing what other kids his age would do, whether that's joining a soccer team or playing baseball, like I mentioned. And so from a very early age, I knew something wasn't quite right, quote unquote. And it took years for me to realize the gravity of the situation and how critical and how fatal this disease was. At the time, I knew my mother had 3 brothers, all of whom passed away from a disease. But, as a kid and as a young teenager, it's hard to really conceptualize what that means and the impact of that. Now, as I grew up and moved into high school and then undergraduate, I became very, very aware and very conscious of what was going on. And at the time, I knew I wanted to do something to fight back. But, again, as a high schooler and as, an early 20-something-year-old, what that materializes into is not quite clear. However, upon and really going through the experience of Harvard Business School, it absolutely helped me to conceptualize and materialize exactly how I wanted to fight back. Before, it was get good grades so I could put myself in a position to fight back. But starting and going through Harvard allowed me to really drill down, expand my network, get out there and meet as many of the researchers as possible so that I could do something. And that something turned out to be the use of customized therapeutics to try and treat Terry, since he really has no other available treatment or therapy, either experimental or approved. And so it was clear that something needed to be done. Through a mentor of mine, Jonathan Fleming, who teaches a great class at MIT. Don't tell anybody that supporting MIT, although coming from Harvard, but I have a deep respect for Jonathan. Jonathan introduced me to a researcher by the name of Tim Yu, Dr. Tim Yu. Dr. Yu is at Boston Children's Hospital. And at the time, this was 2017, Tim was on the cusp of developing the world's first customized therapeutic using an oligonucleotide, or an ASO as it's commonly referred to. And so Jonathan connected the two of us. We had a great number of conversations. And that's really where the effort was born. And so now Dr. Yu's a neurologist, and so we are dealing with a muscle disease. And so it was up to me to try to find researchers in the neuromuscular field who could try to adopt the process that Dr. Yu had used for the little girl with Batten's disease to Duchenne muscular dystrophy. And that's exactly where we started, and that's how things got going.
Now, when we first heard of your story, we were fascinated with how you pulled all of this together, the Cure Rare Disease organization in such an expedited manner. Can you share with us, in broad strokes, the high-level process you went through to plan, finance, and launch the organization?
So it wasn't a meticulous, logical, thought-out plan to get to this phase, right? I, at the time at Harvard, had gotten just one too many phone calls from home where my mom had said that, Terry has fallen. And typically falls for you and I aren't very serious. We get up, we dust ourselves off. However, with rare diseases, and in the case of Duchenne muscular dystrophy, falls can be quite severe, if not fatal. And so after one afternoon in, August, September, when I just got into Harvard in 2016, I was scared, right? I was scared that now I'm very far away from home. I can't be there in an instant. If something goes wrong, it's 7 hours until I can be there. And so that as a catalyst, along with what came after, meeting Jonathan, meeting Tim, meeting this whole slew of neuromuscular researchers, eventually coalesced into Cure Rare Disease. And I had a lot of relationships that I'd formed during my time at Harvard in the first couple semesters, along with those that I'd had in the Duchenne field from just being the brother of a patient, right? And so the beginning of our collaboration started with my brother's clinician, who has been his clinician for now 15 years, Dr. Brenda Wong at the University of Massachusetts. It started with a close mentor of mine, Dr. Lou Kunkel, who taught me an incredible amount about Duchenne, the disease, and the biology behind it. And he's also at Boston Children's. So this small network that I had, I called on these folks to say, let's try this, right? What does it take to just take the first step? I'm not saying come with me on the entire journey. I'm just saying let's take this first step. And this first step in this case was taking a muscle biopsy for Terry. We needed to characterize the patient, in this case Terry, down to both the molecular and genetic level. If we were going to design something that was hyper customized, as Dr. Yu had done for his patient, then we needed to be sure what we were going to design was on target. And so at that point, really a lot of forces came together. The science started to take off the following analysis of that muscle biopsy. The organization Cure Rare Disease was formed, although at the time it was called something slightly different. We started down this road, right? My background isn't a nonprofit. My background, I don't have a science degree. I grew up in a very entrepreneurial environment where we have a problem. Now, how can we solve this problem? And that's the way I looked at it.
Right.
I'm not going to do the science at the end of the day, right? I'm not that smart. But I know people who are really smart in those areas, and I came to know many more people who are smart in those areas. And fortunately, they were willing to take the risk and the effort to join this initiative that could very well change the way that modern genetic medicines are developed. And so, it began simply with a phone call to my mom saying, "Hey, Mom, I want to throw a gala to raise money for Duchenne research." The nonprofit had been formed, and now we were going to try to raise some money. And I had no idea how to throw a gala, no idea how to raise money, but you just tried, right? And the worst you can do is fail. And sort of looking more broadly, we know that the worst that's going to happen without our intervention is already the worst. And so anything that we do will make the situation better, not worse. And so our downside is pretty low, actually, but our upside is huge. And so therefore, it begs the question, how couldn't I do something?
It's a smart way to look at it. And on that note, we at Trial Site News, we recently interviewed someone from Global Genes and learned that there are 30 million Americans with rare diseases and 400 million worldwide. And so as it turns out, rare disease isn't really that rare. And it turns out that there are very few cures for rare diseases, and yet there is such a massive need. Did you come to this realization yourself?
So once I started to get down and into the details of nonprofit development of rare disease, of the biology of Duchenne and related diseases, it struck me quite as you just explained it, that there's a lot of rare diseases out there, right? You cited a great figure. Of those rare diseases, only 5% have effective treatments or cures. So we're saying 95% of the rare disease population has nothing. And my brother falls into that category. And I always like to bring these conversations down from sort of the high level numbers to what does this mean on the front lines? And what this means is you're born with a rare disease with no treatment or cure. The best you can hope for is a decent standard of care where you can maybe have a decent quality of life. Well, that's a pretty bad deal. And so with that number of rare diseases, many of which affect children, we need to do something. And I think the current pace of innovation for larger diseases is great. We're seeing strides in cancer, we're seeing strides in larger diseases, but rare disease needs the focus, because just because you're diagnosed with a disease that you may be the only patient with that disease doesn't mean that you shouldn't get a treatment. And so the question that it eventually drives towards is, how do we design a system or a methodology or an approach that can tackle rare diseases with even a single patient in time enough to save that patient. I think industry as a whole has made amazing strides. We see the work that's being done in COVID. We see the work that's been done in other immune oncology approaches. But rare disease, for the most part, has not had a significant amount of treatments commercialized. And so, I was at that point, one of the reasons I went towards the nonprofit was, it allows us to do science at the end of the day. And our very bottom line isn't dollars raised, isn't return on investment, it's number of patients treated. And to me, that's the most important thing, right? I think society still lacks a significant mechanism by which organizations like ours and others that are coming along can fundraise to direct this capital towards very promising research done in very novel ways. And that's what we're trying to do. We're trying to take this approach of developing a single patient treatment based on CRISPR and show the world that it can work. And it can work not in 10 years and $2 billion, but it can work in around 2 years. And that's really what we're after. And I think by showing the world that we can fix one, then that really opens the door to fixing many. We want to enable this process, this ecosystem, so that anywhere from a single clinician in North Dakota to the largest biotechnology company in the world can both play in the single patient drug development space. Now, there's a lot that needs to be done before that. Don't get me wrong. We've had conversations with payers to try to get them familiar with reimbursing parts of our process, parts that they're familiar with. We've been very active in the regulatory space. In fact, in early March, I went to a CBER session at the FDA where the entire conversation was on the individualization of drug development. And so there's a lot of thought behind this, and we're trying to bring this ecosystem together: reimbursement, regulatory, science, the clinical aspect. Also, we can make this work for not just us, right? At the end of the day, I'd love for other nonprofits to step up into this space, for other hospitals and large pharmaceutical companies to come into the space, because at the end of the day, everybody with a disease needs a treatment. And the question is, how do we get there? And this is one way I hope that we can use and show society is available to treat even a single patient.
Agreed. Now, you mentioned coronavirus. So what are your thoughts on COVID-19 and how it's impacted those with rare diseases? We have plenty of stories that there are many children that are put in jeopardy, for example, those in clinical trials that actually provide the care. For many of these children, the trial is their care, and many of the trials have been put on hold because of this pandemic. Are you finding these kinds of situations to be happening?
Absolutely, across the board. I mean, first and foremost, a big thank you to our healthcare workers and retail workers on the front lines that are dealing with this every day. I think they hopefully won't be the unsung heroes of this crisis. But what we see on the news is heartbreaking. Earlier this morning I was watching again the New York report of the number of fatalities today. It's heartbreaking. But what you don't see on the news is equally as heartbreaking. What you don't see on the news is scientists' labs being closed because of lockdown. You don't see on the news is clinical trials, to your point, of being postponed or delayed. We're fighting a battle where time is our biggest enemy, and when something like COVID, plagues humanity for a certain period of time, everything halts, and that's heartbreaking, right? For us, our example in this case, we've been fortunate in that we've been able to continue to advance the science, because at the end of the day, the science is what's most important to me, right? The other stuff is great, but at the end of the day, science is going to win this battle. So before this crisis started, we always have a plan B. We always have a contingency to fall back on in case something crazy happens. In the beginning of March, when this was entering our radar, we were planning to start our exploratory pilot dosing study. In other words, learning how much of this drug to give based on efficacy results we see in humanized Duchenne mice models. So at the time we said, okay, well, just in case this gets crazy, let's have a contingency to host this study, not at Yale where it was initially to be hosted, but instead at Charles River Labs, because they have operations in place for continuity of business, I believe it's called. And so fortunately, or unfortunately, we fell back on that contingency, and the study starts less than a week from today. And that's amazing, right? The flexibility and the leanness of Charles River to be able to pivot quickly to accept our study, which is critical, right? This study is, upon its success, will kick off everything. And from there on, it's really pedal to the metal, more so than it even is now, to get into the clinic. Where it's been particularly challenging, and I think most nonprofits would raise this and would like to shine a light on it, is in fundraising. We've obviously rely on a number of in-person fundraisers, and a number of those fundraisers, if not all the in-person fundraisers, have been postponed until whenever this clears up, which is hopefully soon.
Wow.
We've fallen back on other options, and it's really forced us to how can we do creative fundraising in a time where people are scared. There's a great degree of uncertainty. There's a lot of moving pieces. And so we're increasing our efforts in advancing gaming as a fundraiser. We've partnered with a few gamers now, and our plan is to host streams. And it's particularly on brand for our audience. Followers and our people we drive and live and hope to cure are those that have physical limitations. They can't necessarily collectively go out and play basketball, as my earlier reference was. And so gaming is something that puts everyone on an equal playing field. And so for us, we see it as a great way to recognize those impacted by rare disease, to celebrate them, to raise money, to advance treatments and cures for them, all while doing what gamers were going to do anyways, which is game. And so initiatives like this are starting to prove successful, and we're continuing to advance them, and we'll continue to advance them after this, right? It's a win-win situation where our gamers get the recognition of really adding value and helping to alleviate some of society's most deadly diseases, and we get to continue to advance the work that we do day in and day out. And so yes, it's unfortunate what's going on, and I hope it soon comes to an end, but we will continue to advance come hell or high water.
And it's very forward thinking of you, using gaming as a platform. So let's go back a little bit here. We talked about some of the connections you've made. So we know from your story that you connected with some impressive researchers and scientists that have helped you get the research efforts going. Can you share with us the contribution they've made, and how has finding the right scientific and medical expertise at the right time helped you to progress?
Yeah, absolutely. So there's a couple things there. I sort of look at it through the lens of finding the right researcher is sort of like Goldilocks, right? Not too hot, not too cold, just right. Our ideal researchers are those that are the smartest in their field, willing to collaborate, who see the vision and want to collectively move towards that vision, and who are willing to venture into the great unknown. A close mentor and colleague of mine, Dr. Lauren Black at Charles River, uses the analogy oftentimes, and I hope she doesn't mind me borrowing it, of clearing the forest. Right now, we're at the edge of the forest, and the science that we're doing is clearing but a path through the forest. This science will clear one path through the forest that hopefully others can follow. And so you can just tell when you have the right team. Things start to flow. People start to move forward and take initiative to advance their specific action item and understand how their input affects the whole. So for instance, we've partnered with the University of Massachusetts to host the clinical trial. And then for a while we debated this for a while to understand where's the best place to host it. And with the guidance and support of Dr. Brenda Wong at the University of Massachusetts, really one of the preeminent, if not the top neuromuscular Duchenne clinician in the world, and also a great friend, right? She's cared for my brother for 15 years. And so there's a relationship there. At the end of the day, this all comes down to relationships. Getting people to do what we need to do and to work together will end up helping us win this battle at the end of the day. Another researcher that I'd love to call out is Dr. Munkle Leck and his team at Yale. Dr. Leck, a patient himself with limb-girdle muscular dystrophy, understands not just the scientific perspective, but the patient perspective. He takes careful care to make sure that we minimize anything that touches the patient, any risk that the patient needs to take. And he and his team drive forward. They literally work through Christmas and New Year's to get our therapeutic off into preclinical development. And that's saying something. It's not about the money. It's about the patient. And beyond the researchers and clinicians, I've named just a few. There's a whole number of more whose names I'd love to just run down and highlight every accomplishment. I don't think we have time for it, but it's a team. And beyond the researchers and scientists are the folks who are helping us make this happen. This is ranging from the mom in California who makes her $10 donation every month to one of our global philanthropists, Len Blavatnik, who established the Blavatnik Fellowship at Harvard Business School and is the reason that I was able to start this. His support and care towards the life sciences allowed me to take the risk of diving into this. And so it's an entire equation. It's a system that's come together to help us get here, from our researchers at Yale to Charles River to the University of Massachusetts to other organizations that we've now partnered with to help with manufacturing, and really painted the trail to the finish line. We've surrounded and have been fortunate to be surrounded by a group of people who are passionate, who understand, and really want to do good in the world, not just because there's a financial metric attached to it. So it's a complicated equation, and you hear this time and time again, it's all about the people, and I'll stand here and say and reaffirm that, again, it's about the people.
So you mentioned earlier about customized therapy. Now, our audience, the Trial Site Network, is a large and diverse group representing many different interests. Could you break down this concept so that as many people as possible can understand the concept? And also, if you could share if this is the same concept as personalized medicine or precision medicine.
Sure. So the terms personalized and precision, some use them interchangeably, others use them as standalone terms that mean different things. I'll define what we're doing by describing it. And whether I call it, or we call it, customized therapeutics, others call it precision medicine. I'll let the people smarter than me sort of sort out the nomenclature that should be used here. But basically, at the end of the day, each of our bodies have over 20,000 genes. Genes are made up of different components, including exons and introns. And the difficult part with genetic diseases is that a disease, for instance, like muscular dystrophy, is a blanket term that encompasses a number of muscular dystrophies. You've got Duchenne, you've got limb-girdle, you've got myotonic, you've got a whole list of diseases under muscular dystrophy. Within Duchenne, there's subsets of the population there. So you've got, the dystrophin gene is made up of 79 building blocks or exons. And problems can occur in any one of those areas that cause disease manifestation. And so in my brother's case, his exon 1 is mutated. In another patient that we're working on, that patient has a duplication of a set of exons from 20 to 25. And so what we're doing is designing therapies specific to the individual. Even within an exon 1 mutation, the breakpoints are in different areas, which could call for a different type of approach. And so you start to sit back and go, wow, okay, a rare disease in a rare disease in a rare disease. You're like a rare cubed disease at this point, where you may literally be the only person in the world with that specific mutation. And so it begs the question, how do you treat an entire population with a one-size-fits-all therapeutic? And so far for my brother, the answer has been, well, you don't. That being an unacceptable answer to me and the millions of families like me, we started down this path by trying to develop something very specific for Terry and his mutation, and to take those learnings and apply the infrastructure we've now set up, from the discovery lab to the preclinical studies, to the clinical studies, to the manufacturing and regulatory partners. See, that whole infrastructure is now set up, and it was set up for one person. But now we can take 2 and 3 and 4 and 5 and 6 and n different number of people and run them through that same process. But at the end of the day, that's why the analysis that I mentioned earlier in our conversation is so important. The whole genome sequencing, the RNA sequencing, which all comes from, in our case, a muscle biopsy, because it's a muscle disease. And so it's really important to identify the very specific mutation, even just beyond the exon. Where are the breakpoints? Because that allows us to design something that will fit. Think of it as a lock and key type of method. We're trying to find the specific lock, we're trying to find the specific key that unlocks the disease-causing mutation and helps to improve the patient. So even 2 patients with Duchenne muscular dystrophy, again, will possibly have 2 different keys that will be the solution to their treatment. And so that's what we're trying to prove here, is that A, it is possible to do this. B, we can enable the ecosystem from, again, regulatory, reimbursement, science, clinical development, all this, to come together to treat these patients. And so when, yesterday Monkul and I had a great conversation with another possible patient in our pipeline. And so we, ran whole genome sequencing on them or on their son, and devised a strategy to treat that disease-causing mutation. In this case, again, it's Duchenne. And so what we're doing down to the very specific guide RNA, down to the specific cutting strategy of the gene using CRISPR, is what we define as customized medicine. Think of a tailor-made suit. It's designed for you.
So, on the topic of actual medicine and your brother, how is the development of his treatment coming along?
So it's coming along quickly, quickly, efficiently. Every day is a challenge. Again, we're sort of clearing the path through the forest. And so there's a lot of unknowns and things that we run into that, nobody necessarily has an answer. And, in some regards, that's scary, meaning that nobody's answered this question in the world before. So how can we take other learnings and apply it here? But overall, we and I have found the biotechnology community to be a friendly one, to be willing to share and talk and collaborate, even if they're working at other companies on similar projects. And to drill down into more of the specifics with Terry, we took his muscle biopsy in late 2018, conducted the bioinformatic analysis on it. That's whole genome sequencing, it's RNA sequencing, what's going on with the DNA, what's going on with the transcript, the RNA, and what's going on at the protein level. At the end of the day, the protein's the most important. And to develop something specific, we need to understand where is the error happening. And so by April, the team at Yale, having been supported by the team at University of Massachusetts where the biopsy was taken, UCLA and the Nelson lab, who conducted the bioinformatics, had shown in vitro or in a dish proof of concept at correcting Terry's cells. Since then, the therapeutic was finalized and we started preclinical development a couple months ago. Our bioinformatics, or our biodistribution study, is being completed. And that's allowing us to see, with this construct, where did it go in these humanized Duchenne mice? And was there a dose-dependent response? And so that question's been answered. Now, what's really exciting is our first dosing study, what we're calling our exploratory PD study, will begin in less than a week. And there we'll get to see, not even just in Terry's cells, where we've shown proof of concept, but now in a living organism, can we upregulate the missing dystrophin, in this case cortical dystrophin, that's needed to help the patient. And so this is a nail biter, right? Every day is one of those where we've got a million moving parts and they all need to be in sync. And so a little bit about the therapeutic. It's an AAV therapeutic, meaning it's a CRISPR-based therapeutic mediated by AAV, meaning that it's a dead CRISPR inside an AAV with a muscle-specific promoter, because we only want this to express in muscle, and we don't want dystrophin to express in an eye or some other off-target tissue,
right.
Along with a couple other components. And so, the idea of testing pieces of these components through biodistribution, we test the AAV, we test the muscle-specific promoter with a green fluorescent protein. Now moving on to dosing, this is the actual therapeutic that would go into Terry.
Okay.
And so, things are moving along, right? We were fortunate to have this contingency that I mentioned. And so Charles River will start the exploratory PD study next week. And that'll be a 4 and 8 week study where, we're hoping to see increases of not only transcript, but also protein in these mice. And to be able to measure those and really say, okay, at this dose, this was the corresponding increase that we saw in the protein. And so from there, we'll move on to a larger dose finding study, again with Charles River. And then we'll close out the last of our studies with a GLP toxicology study. Basically, how much do we give, what we learned in the dosing studies, and what is the safety profile at that dose. And so we're doing a lot of things in parallel to be able to save time, because again, time is really the thing that we have the least of, and trying to be really creative about our collaborations and who we work with, and do they get the message and do they sort of buy into the vision? So it's a complicated equation, but fortunately we're moving forward.
So we know a little bit about the science behind your development approach. We believe, as you mentioned, you were using CRISPR technology, gene editing. Could you share more for the layperson how the science works?
Sure, happy to. So imagine that you've got this ladder with 79 steps, each of which you need to go from step 1 to step 2 to step 3 to step 4. Right. So in Terry's case of his ladder, he's missing the very first step, which by that logic should mean that he produces little to no dystrophin. And so our job is not to skip over a step, because we can't skip over the first step. We need to upregulate or activate that first step of the ladder so that the rest of Terry's innate infrastructure can produce the protein that he so desperately needs. So the technology we're using, yes, is CRISPR. Traditional CRISPR, as we have come to more or less know it, is knocking out an exon to allow for read through. We're not doing that here. We're actually doing something a little different. We're doing what's called CRISPR activation. We're trying to upregulate the alternative form of his dystrophin gene, which we found to be trace elements present when we did the muscle biopsy. Again, very important to start at the beginning and understand everything we're dealing with, to be able to allow him to produce full-length functional dystrophin. And so the way that CRISPR works is that it basically homes to the specific cut site or the mutation site. In this case, we needed the guide RNAs, which are part of the CRISPR therapeutic, to home to the mutated exon 1 area. Once they're at that area, then the other componentry of the therapeutic, all commonly known as CRISPR activation, starts to induce the upregulation of this alternative isoform, this dystrophin protein. Not to get too technical, there's the primary form of dystrophin is the muscle isoform. That's the mutation that my brother has that doesn't allow him to produce it. The other two aspects of dystrophin, the primary isoforms are the Purkinje and the cortical isoform. And so our goal, based on evidence that we saw in the muscle biopsy and the analysis that happened afterwards, is that this would be the best approach for Terry to be able to have not only skeletal muscle improvement, but also cardiac muscle improvement. Because what happens with these boys and young men is that oftentimes one of the last things to go is the heart. And of course, we all know that we can't live without that. And so, our goal is to show functional improvement of the cardiac muscle, as well as a degree of functional improvement of the skeletal muscle. Our expectations for each of these customized therapies that we are and will work on are different based on when we, quote unquote, catch the patients. So obviously, the earlier we catch it, before a significant amount of the disease has manifested, a significant amount of scar tissue and other stuff like that, bad stuff like that happens, is that we can, again, the younger we can catch them, the better we can avoid some of those effects. So, for Terry, our hope is that we restore a degree of independence and upper limb mobility and strength and heart function to him. Whereas, another boy we're working on who's 4 years old, the duplication I mentioned earlier, our expectation for him is that in all likelihood, if this works, he'll never even know that he had the disease, which is a powerful thing to say. And really, Terry's risk and sacrifice here will help pave the way for other patients that come. We're not just fixing Terry and done. We've got a whole cohort of both Duchenne boys and young men, as well as a few limb-girdle muscular dystrophy patients now that we're working on. And so, again, taking this from the single patient to, how do we scale this up into 5 or 10 patients at a time, and getting closer to what industry recognizes as familiar territory.
So you, at the beginning of this, when you talked about how you got started, your first gala, you weren't even sure how to do it, and you've come a long way since then. So how big is your organization now? We know that you have put some partnerships together. Do you have all the pieces in place to get the first customized therapy approved, or do you anticipate a number of additional steps? And if so, how many?
So I'm proud to say that as of today, we have all of the necessary infrastructure in place to get this from where it is now in preclinical development to the infusion into the patient, which we expect to be either late this year or early next. So we've got the manufacturing partners lined up, our clinical partners lined up, our regulatory support services lined up, and we've got a plan that we are all aligned on and marching forward to the tune of. And so, it's taken a lot of work, but I'm very glad to say that the infrastructure for not only this case is in place, but future cases as well is now something that we can do. So, the road here is a treacherous one and a tenuous one, but we are making progress on it.
Would you have to go through formal phase 1 through phase 3 clinical trials for this class of customized therapy, or are there expedited processes that the FDA can work with you on?
So the FDA is becoming a much more nimble and creative organization than in years past. With the development of these next generation technologies, whether it's gene replacement therapy or CRISPR therapy, the FDA is learning at a similar pace as industry and society is. And so our interaction with the FDA has been positive. Our interaction with the FDA has been helpful to guide us towards and get feedback on critical experiments and also unnecessary experiments. And so to answer your question, yes, we'll need to get FDA's approval, which I fully support and thinks that's very necessary for the well-being of public health. But we won't have to do the typical phase 1, 2, 3 type clinical trials. And the reason for that is we're taking a path called expanded access individual patient IND, or investigational new drug. Basically, it's an initiative or a regulatory pathway within the FDA that allows for expedited and curtailed testing when it's a customized treatment for 1 or 2 individuals. It's very small scale, and that's really urged by the necessity of the patient, right? So if you got a very serious clinical outcome where we already know what the outcome is going to be if we don't take action, which in this case we do, the FDA will work with you to streamline and expedite both studies and approval processes so that we can get to the patient. Above all, the FDA wants to make sure that you do no harm. And so a lot of emphasis is put on making sure the therapy will be safe, right? In our case, do we cause unintended off-target effects? And we can test that. Other components that we need to prove safety for. And whether it's a regular IND or in our case, an expanded access individual patient IND, the FDA will require safety testing. And belief that the therapeutic and the risk of taking it will have some benefit to the patient. So efficacy in this case. So to sort of answer your question and summarize it all up into one sentence, no, we won't have to do the typical phase 1, 2, or 3. Yes, we'll still have to do safety and efficacy testing, which I'm fully supportive of. And fortunately, we'll be able to move quicker than the traditional clinical trial or the clinical infusion because of these regulatory pathways.
So for those thinking about following in your footsteps and getting into biotech to help a family member or someone they care about, can you share some of your lessons learned?
Yeah, absolutely. I was thinking about this just the other day, actually. Again, I don't have formal biological training. I don't have a PhD, an MD, or anything like that. I think what's allowed this initiative and this project to succeed so far and to continue to do so is a couple factors. The first is the team, right? It's an area where teamwork and surrounding yourself with the smartest, the most collaborative people will pay massive dividends. So the team is a very important part. Find the right people. And that means talking to a lot of people. I think I don't know how many conversations or, coffee chats or one-on-ones I've had, but probably in the thousands. That grinding through and getting to know people through those conversations and building those networks has been invaluable, because now I can say, okay, well, who's a virology expert? Okay, well, so-and-so. Okay, great. So those conversations and that networking has allowed me to build a sort of a Rolodex of people that are with us to varying degrees. The other thing I would say, and this is something that Harvard really does a great job at the business school, is ask questions. And when I Harvard promotes this a lot to say, ask questions. And when I first heard it, I sort of interpreted it as, ask questions out loud to the individual presenting. And sort of what I've realized over time is it's not necessarily just ask questions out loud, but ask questions to yourself. How does this work? Broad questions, right? Within any system, whether it's biology, whether it's manufacturing, whether it's consumer electronics, there's the question of, okay, what's the problem? What's causing the problem? And how could we look at fixing the problem? And those are quite broad, right? And so that's sort of the early questions that I remember asking myself. Once you get into sort of the weeds, or once you get down a work stream, these same questions still serve you well. How do we get access to hosting this clinical trial at a university? That'll unlock a whole slew of other questions. Well, you need to do this, you need to do that, you need to do this, you need to get your clinical trial agreement, you need to get your IRB approval, you need to get your clinician on board. Okay, so how do I do each of those? And again, not necessarily asking an individual, but asking yourself, because once you have internalized the questions that you need to ask, and more importantly, the answers that you need to get, you've got your path. Okay, I need to get manufacturing. Or taking a step back even further and saying, okay, how do I build a CRISPR therapeutic? Well, I need this and that component. Okay, so who's the experts in those fields? Okay, let's talk to them. Now we've got the construct in an academic lab. What do we need to do next? Okay, we need to do preclinical testing. Okay, who does that? How do you do that? Again, these questions of who, how, what, it's the same question, just in a different shape or a different application. But nonetheless, asking the question, even though you may not have the answer, exposes the need to find the information that answers that question and helps build the guide for what will be the entire project. And so, the questions I'm asking myself now are, how do we speed up this study? How do we uncover this uncertainty? How do we get more understanding about CRISPR immunogenicity, for instance? These types of questions, again, what, how, and where, are really effective at guiding the project. And so I don't have the answers to most, if many of these questions, but I know people who do, or I know people who may not have the answer, but have done something similar, and that we can pull them into the collaboration and benefit from their knowledge, even if it's not apples to apples, right? CRISPR is not apples to apples with gene replacement therapy. And so, but people that have worked on AAV delivered gene replacement know how AAVs operate. And so we can take those learnings and apply it to CRISPR, because again, we're delivering by AAV. So you see, I think it comes down to team, the questions, and really risk taking to be able to dive into the unknown and be confident enough about oneself and one's ability to learn as a team, learn as an individual, to continue moving forward. I remember when I first started this whole thing, I read a lot of PubMed papers, and I got to say, I probably understood about 20% of what was on the page, but it was a start, right? At least it allowed me to ask more questions.
Now, Rich, we know you're a busy man, but before we let you go today, is there anything else you'd like to add for the viewers?
Disease is a very terrible thing. Disease is something we oftentimes can't control. Disease oftentimes just takes us when we're least expecting, or impacts us when we're least expecting it. And the thing I'd want to get across to viewers is, you're not alone. I remember growing up in, very rural upstate New York, and I didn't know of a single other family who had Duchenne muscular dystrophy. And at the time, I just took it as my normal. But I think encouraging people to think outside the box, to know they're not alone, to know that there's somebody else similar, if not in the same shoes, that are going through something just as terrible. It's not an answer, but at least it brings you some solace and hope that there could be a better tomorrow. The courage to reach out to an investigator to say, hey, my son, my daughter, my loved one, my brother, my mom, my dad has this disease. You're working on it. I'd love to talk. You never know where these conversations will go, right? This is a long and very painful and very arduous path to take. But if nobody does it, then we'll never have a better answer for tomorrow. And so there's a reason why Duchenne has taken so long to treat. And it should have been fixed years ago, but it's not. And so I encourage people to think outside the box, to not only hope for a better tomorrow, because I find hope to be a bit of an intangible that oftentimes is more troublesome than it's worth, but to take those steps to try and change tomorrow. Right now, my family and I are paying the pioneering costs, so that families in the future won't have to suffer through this. And, I encourage people to reach out and to learn more. Our website, I suspect will be provided on this podcast, is cureraredisease.org. People interested in partnering and working with us and volunteering, we welcome everybody. Everything from even a dollar donation to, a large global philanthropist, everything makes a difference. You never know who in your network that we or anyone could talk to that can make a significant difference. And so, I'd close by saying, I really appreciate the opportunity to come on here and speak and just share a perspective. I don't know if it's the right perspective, but it is a perspective, and hopefully one that helps to alleviate suffering down the road.
Well, Rich, we're very grateful for you taking the time. You're an inspiration, and we wish you all the best of luck in building a highly successful organization and getting a cure for your brother.
Thank you so much. Thank you so much. Thank you so much.